Good afternoon, everyone, and thank you, Jason, for the introduction. As he mentioned, today's webinar is going to talk about how we investigate angiogenesis in an in vitro setting. I will be talking today about our two human co-culture approaches to investigating angiogenesis that we use in conjunction with our live content imaging system, the IncuSight Zoom. So you may have heard me just mention the term live content. What do I mean by live content? I'll go over this first before getting into the angiogenesis portion of the talk. We define live content imaging here at Esen as the acquisition, analysis, and quantification of images from living cells that remain unperturbed by the detection method, allowing for repeated measures over long periods of time from days to weeks. And what I'll show you today in our angiogenesis assay is that we can image our assays anywhere from 5 days up to 2 weeks without any perturbation of the cell layers. For more in-depth discussion about live content imaging, I'm showing you a Venn diagram where live content imaging highlights the special aspects from multiple different techniques used in biology to investigate cells, namely live cell imaging, high content imaging, light microscopy, and plate reader simplicity. Focusing on live cell imaging first, we consider live cell imaging the spatiotemporal real-time resolution of cellular phenotypic changes. And what this allows us to do is take snapshots in time of our cell population over the course of the assay. So for example, in our angiogenesis assay, the data I will show you today from our assays is we take temporal updates every 6 to 12 hours over the time course of the assay to generate our kinetic data. From the images that are collected, we can generate image collections. We can also generate movies to look at how our cells change over time. And we can also, as I mentioned, do repeated measures where we can set up our assay from the start and do initial time course experiments without having to determine where that endpoint is going to be. We can retrospectively go back, choose an endpoint data, and do some more in-depth analysis at that time point. In terms of high content imaging, when you think about high content imaging, you think of multicolor automated approaches to investigating cells at certain time points. In our system, it's a 3-channel system where one channel is phase contrast imaging, and we also have 2 fluorescent channels, green and red fluorescence. The image analysis and acquisition is fully automated once you put your cells into the incubator, and that's another point, that the cells remain in their native condition in an incubator throughout the course of the assay. And once you put your cells in the instrument inside an incubator and tell the instrument when you want to image and how many updates, your time course of your assay, the instrument does the rest of the work. It will go out at those time points, image your cells, and repeat and send the images back to your system. From that, you can do quantitative plate-based pharmacology to look at pharmacological responses over time. In terms of light microscopy, we've worked very hard to develop high-quality phase contrast images to look at our cell populations over time and design this for researchers at all levels. So we can use the instrument to do simple things such as monitoring tissue culture cells over time to look at changes in morphology, changes in cell population, as well as doing more complex pharmacological studies and mechanism of action studies in looking at cell proliferation, cell migration invasion. Some of our newer assays include neuroid outgrowth, and what I'll be talking to you about today are co-culture-based angiogenesis approaches. Also, we've designed our software to be very intuitive and simplified, such that a new user can pick up the technique very quickly. It's very intuitive software interface that allows easy analysis of cell populations that can be accessed either from your computer at your desk, or even if you can VPN to your network, you can access from home. So you can look at your cells whenever you would like to see how they're progressing in the experiment. And we put this all together in a single package called the IncuSight Zoom. And the IncuSight Zoom is a fully automatic 2-color phase contrast and fluorescent microscope designed to work inside a standard tissue culture incubator, and is ideal for kinetic, long-term live content imaging. So here in the upper right, you see the IncuSite Zoom, the imaging portion of the instrument that is sitting inside of a standard incubator, along with other flasks that you can use to carry your cells. The Zoom is diverse in that it can image anything from a T25 flask to T75 flask, as well as up to a T150, T225 flask, and even tissue culture dishes, 100-centimeter tissue culture dishes, and microplates, anything down to from a 6-well plate down to a 384-well plate. Regardless of brand, we try to make it diverse so that you can image multiple brands of plasticware and multiple modes of vessel. For the purposes of the angiogenesis study and angiogenesis assays that I'll be discussing today, these are both 96-well-based assay formats. And I'm showing you here an image from our prime kit, our primary cell-based model, where the cells have differentiated into tubes. The endothelial cells are differentiated into tubes, and we can image that in the IncuSite. And I'll show you how we get to this point and how we quantitate our tube formation over time by the end of the assay. Or by the end of the webinar. I will then go in to discuss how we generate pharmacological responses and use this to investigate physiologic relevance to in vivo pharmacology. Just some background on angiogenesis in general. Here I'm showing you a summary of the angiogenic process specific for cancer pharmacology, but this also applies to other aspects of physiology such as diabetic retinopathy, rheumatoid arthritis, and even such normal physiological processes as what happens during wound healing. In terms of tumor pharmacology, tumor biology, a tumor, once it reaches a point in growth, needs to have nutrients supplied to the tumor for further growth. So it will release factors, growth factors such as VEGF, that will then communicate with the existing vasculature. This will cause an activation of the endothelial cells. The endothelial cells will then proliferate and begin to migrate toward the tumor environment. This involves the breakdown of the extracellular matrix, followed by differentiation and growth of these endothelial cells toward the tumor. Finally, the endothelial cells will begin to connect and form tubes, and as well as form a lumen to support blood flow and recruit smooth muscle cells, including pericytes, as well as stromal cells, such as fibroblasts, to strengthen and stabilize the vasculature, resulting in a mature vascular bed. This then allows the tumor to grow and eventually potentially metastasize to other tissues. Now, in designing an in vitro angiogenesis assay, it's important to consider the microenvironment in vivo, where we see, in the case of breast cancer, that multiple growth factors are involved in promoting angiogenesis. And why is this? What is the reason for having multiple growth factors? Well, it could serve as a protective mechanism for the tumor to protect it from chemotherapeutics. It also might be responsible for the development of resistance to chemotherapy. And we see this in the case of VEGF, where anti-VEGF therapies, such as Avastin, are very effective in targeting the VEGF signaling response, but we know that the tumors then develop resistance and eventually overcome the anti-VEGF treatment. Finally, these growth factors may be involved in the development of heterologous tumors within multiple, resulting in the development of multiple tumor cell types, such as cancer stem cells, again, promoting tumor survival, developing resistance, and eventually metastasizing to other tissues. So it's important to us to have a system that responded to multiple growth factors, not only VEGF, but outside of VEGF, such as basic FGF, TGF-beta1, placental growth factor, or even EGF. And I'll show you some of those data in a second. First, just to talk a little bit more about our approach, we've developed two characterized and highly validated in vitro assays to measure angiogenesis in real time, and we call these the prime kit and the stem kit. The prime kit is a mix of human umbilical vein endothelial cells and normal human dermal fibroblasts, while the STEM kit is a co-culture of stem cell-based cell lines, endothelial colony-forming cells in co-culture with adipose-derived stem cells. Now, when you grow these cells in an unperturbed state, when they're uninfected, it's impossible to distinguish the tube-forming or the endothelial cells from the stromal cells or the ADSCs. So we had to develop a method of being able to visualize the endothelial cells, and we do this by using a non-perturbing lentiviral infection where we transduce the cells with a cDNA encoding green fluorescent protein, and this allows us to visualize the endothelial tube-forming cells while and differentiate them from the underlying stromal cells. When we set up our kits, we place them in the IncuSite Zoom, and where we image them inside the incubator in both phase and in the case of angiogenesis, it's a single color experiment, so we image them in phase and green fluorescence using either a 4x or a 10x objective. And this is the type of image that is put out that you see in the third image from the left, where we can measure the cells, and this has the segmentation mask where our custom angiogenesis algorithm has been applied to differentiate the tube-forming events from non-tube-forming events and quantitate the tube formation over time. And that's shown in the upper right graph where we're looking at a VEGF concentration response curve. So when we apply VEGF at different concentrations, you can see an increase in tube length over the course of the experiment. And this experiment has run for about 4 days, where we see tube length reaching levels of 11 to 12 millimeters of tube per area. It's important to note we normalize all of our data to image area. That normalization to area that you see is to image area, to be able to cross-compare across different objectives, so that we can compare data taken from a 10x system to data taken from a 4x system without doing any other manipulation to the data. How do we make this quantitation? Well, here I'm going to show you a series of images taken over time in our STEM kit. Here we're looking at ECFCs at time 0. This is taken 3 to 4 hours after the ECFCs have been seeded, and it is taken at the time of growth factor addition. This culture is treated with 20 nanogram per mil VEGF, and I will show you how the networks develop over time. So at 12 hours after growth factor addition, you can see that the ECFCs start to elongate. You'll see that there's some of the cells start to look like tubes, and by 24 hours you can see the networks are really starting to form, and these tube structures are starting to take off. If you look 2 to 3 days later at 96 hours, you see that the networks have developed. They've become well established at this point, and you have a vascular network formation using the STEM kit. We then apply our custom angiogenesis algorithm, which goes through these images line by line and pixel by pixel to differentiate foreground fluorescence from background fluorescence, and then creates a segmentation mask, which we can then apply filters to filter out non-tube forming events from tube forming events. And here we're looking at the white area of the mask are the tube forming events, while the blue are areas that are filtered out that we've told the algorithm are not non-tube forming events. The algorithm will then apply a single pixel line through the middle of the segmentation mask, and we know the length of that line, and it will generate data such as tube length, tube area, and branch point formation that we can use to assess the angiogenic potential of a growth factor or an inhibitor. Here you see that our tube length metric increases over time from 2.8 millimeters of tube per area to 12 within the first 12 hours, and then there's a period of refinement and smoothing of the networks to resemble a vascular bed by the end of the assay. We also receive the kinetic output from this that you see in the lower left, where you can see the snapshots in time of where these points lie along the curve, and we've taken 6-hour updates using the STEM kit treated with VEGF, and you see the tube length measurement over time. The next question is, why did we decide on a co-culture approach? What's the importance of co-culture for us? When we were developing our assays, we wanted an assay that closely resembled the in vivo network, as well as being able to correlate highly to in vivo pharmacology. We considered the Matrigel approach, but the cohorts that form there, we feel that the Matrigel approach measures mainly migration and invasion of the cells that are involved, as well as the cord networks that are developed can develop with cells that are non-tube forming cells in vivo, such as fibroblasts, pericytes will all form these cord networks in a Matrigel assay. And that didn't seem to be the most relevant approach for us. So we focused on the co-culture assays. Our initial assay was a prime kit, combination of HUVEC and fibroblasts, and we've since developed the STEM kit, a combination of endothelial cells and ADSCs, where these networks form over time and closely resemble the in vivo network, which you can see a rendering in the middle image. And you can see how closely our networks resemble that rendering. Furthermore, when we looked at the pharmacology of both the prime kit and STEM kit, the pharmacology of these assays correlates very nicely with published and developing in vivo pharmacology, and I'll get into some of that toward the end of the webinar. So here's a side-by-side comparison of our two approaches. I've already spoken to the cell types that are involved in our STEM kit and prime kit. Both of these kits are available as cryopreserved kits that we would ship to you, and you would set them up in-house. Or you can, if you would like, you can send your compounds to us, and we will be happy to run the assay here in-house as a service. We would generate the data, send you a report at the end of the assay that summarizes the data that were generated, as well as data and images from the assay. The prime kit, because it's a longer assay, can also be sent to you as a live kit where you would receive the cells live, and then it would just be a matter of feeding the cells and measuring angiogenesis over time. A little bit about the duration. The STEM kit's a little bit of a shorter assay, which is why it's not offered as a live kit. It's only in studying tube formation, it only takes 3 to 5 days, whereas the prime kit is an 8 to 10 day assay. The prime kit does measure all phases of the angiogenic process, including proliferation, migration, and invasion, and differentiation and anastomosis, whereas the STEM kit mainly is studying the migration, differentiation, and anastomosis of the developing network. When we look over multiple plates over length of time, we see that the statistics generated by both assays are very similar. We get very similar average tube length on the order of 10 to 14 millimeters of tube per area, with coefficients of variation that are in the 5 to 10% range for both assays. The average Z prime for these assays is 0.5 to 0.8, indicating that these are excellent assays for screening purposes. Further, with the STEM kit, we've found that the ADSCs, and I'll show you data in a bit, that the ADSCs do differentiate into a pericyte-like phenotype and express pericyte markers such as alpha smooth muscle actin and PDGFR-beta, and can be used to study resistance to large biologics such as VEGF antibodies. Finally, the prime kit we've shown does have lumen formation. We've shown this by transmission electron microscopy. We have yet to determine that for the STEM kit. To focus first on the prime kit, I'm first going to show you a movie of VEGF-stimulated tube formation in the prime kit, so you can see how the HUVEC differentiate over time. Here, this experiment was actually started about day 3, so you see where the cells are on day 3, and when I play this, you'll see that these cells are very dynamic. They are continually moving and differentiating, and by the end of this movie, you'll see that the network that forms is extensive and you see a high level of branching and complex network formation by the end of the assay. And you'll start to see toward the end that the networks will join and meet up and form one large continuous network. So taken every 2 hours, these data are taken, excuse me, these data are taken every 2 hours, And this is a very information-rich data set that is generated because we not only see the movie, but we can also get the quantitation of the growth factor response from these data. So how do we do that, and what does it look like? I showed you how our algorithm quantitates pharmacology. And here we're looking on the left at two different growth factor responses, VEGF and basic FGF. What you can see from these curves is that the VEGF and basic FGF response in the prime kit is very similar, such that we see a concentration-dependent increase in growth factor stimulation of tube formation by both VEGF and basic FGF. Excuse me. And they reach similar levels by the end of the assay in this experiment, about 9 to 10 millimeters of tube per area. When we look more in depth at the concentration response at three growth factors shown in the lower right-hand panel, VEGF, basic FGF, and EGF, you see that not only do they show similar potencies with EC50 values in the 1 to 2 nanogram per mil range, but that these data are highly reproducible, showing low standard deviation across 4 separate experiments. But what about inhibition, inhibiting the VEGF response or a basic FGF response? Here we looked at a large biologic anti-VEGF antibody and its ability to inhibit both the basic FGF and VEGF-induced tube formation. VEGF and growth factor and the anti-VEGF antibody were added on day 2 in this assay, and the tubes were allowed to develop over time. And what you see is with VEGF, there's a concentration-dependent inhibition of tube formation with in the presence of the anti-VEGF antibody, where 2 to 4 nanogram per mil results, or 2 to 4 microgram per mil of the antibody results in a complete inhibition of tube formation. However, interestingly, with basic FGF, the story is a little bit different, in that the anti-VEGF antibody really had little to no effect on the basic FGF response, indicating that basic FGF is signaling through a different mechanism and is not as reliant on VEGF for its pro-angiogenic response. What happens if we allow the networks to develop and then introduce the anti-VEGF antibody? Well, that's shown in the upper right, where we treated on day 2 with VEGF, and allowed the networks to develop for 10 to 12 days before the addition of the anti-VEGF antibody. So here we show anti-VEGF addition on day 12, and you can see that at 2 and 4 microgram per mil, we really increase the kinetics of tube regression. So this antibody, the networks remain sensitive to anti-VEGF therapy and enhance the rate of regression of the angiogenic network. So we can study some complex biologies using the prime kit, but I want to focus for the rest of the time on our newer kit, the STEM kit, and how it can be used to model angiogenesis and look at vascular resistance. And this was work that was done in collaboration with a group at Eli Lilly and was recently published in the Journal of Hematology and Oncology. So using our STEM kit, again, it's a co-culture of human-derived stem cells, ECFCs and ADSCs, where networks develop in the first in 4 to 5 days. And you can see that here, where growth factor is added at time 0, and VEGF stimulates tube formation very rapidly and then stabilizes over the next 3 to 4 days of the assay. And using a general tyrosine kinase inhibitor, Suramin, we're able to completely inhibit tube formation down to 0 at 100 micromolar Suramin. As I mentioned, we are also able to look at pericyte development in the ADSCs. And here we fixed and stained the cells for a pericyte marker, Desmin. And what you see is that the ADSCs that are in close proximity to the networks have differentiated and are expressing Desmin. Further characterizing the assay, we show here that in the top panel, that the ECFC expressed the vascular marker, PCAM or CD31, and this speaks to 2 things in our assay. First, you see the GFP expression on the left and the network that's formed in the presence of GFP. and that it nicely overlays in the top right with the PKM CD31 expression, indicating that that our ECFCs express vascular markers, and that using our GFP approach, we are measuring 95 to 100% of the tubes that form in a single well. In the lower panel, we're looking at another vascular marker. This is the angiopoietin receptor, TIE2, and you see, again, nice co-localization of the TIE2 receptor with the developing tubes. I mentioned that the ADSCs differentiate into a pericyte-like phenotype, and I'm showing you more data here to speak to that, looking at two different pericyte markers, smooth muscle actin on the top and PDGFR beta on the bottom. Looking at smooth muscle actin first, you see, again, a nice overlay where the smooth muscle actin expression is in close proximity to the angiogenic network of the ECFCs. You also see that the smooth muscle actin is not present in the spaces where there are no tube, indicating that it is the endothelial cell ADSC contact that is important for the induction of the smooth muscle actin staining. This is similar with PDGFR beta, where the ADSCs that are in close proximity to the endothelial colony forming cells are expressing PDGFR beta, another known pericyte marker. But how do these cells respond to growth factor stimulation? So here I'm going to show you a side-by-side movie of basic FGF on the right and VEGF on the left, looking at the growth factor responses in the STEM kit. So you'll see over the course of the 4 to 5 day assay that both VEGF and basic FGF stimulate tube formation using this approach. And by the end of the assay, you'll see a thickening of these tubes. For example, VEGF here, basic FGF over here, where these tubes become very thick, indicating that they are mature and developing in mature networks at this point. And also speaks to the multicellularity of the developing tubes. Not only do we get the imaging capability of making movies, we can also quantitate the response, the growth factor response, and look at how growth factors are affecting tube formation over time. And shown here, we see, again, similar to the prime kit, we're able to measure a concentration-dependent increase in tube formation over the course of the assay. So you see in the gray line, that's our no growth factor control, where we have a basal level of tube formation, and as we increase the growth factors, whether it be VEGF or basic FGF, you see an increase in tube length over the course of the assay. One thing to note that's a little different than the prime kit is that this STEM kit does require a higher concentration of growth factor, where 10 to 20 nanogram per mil growth factor is a maximal response, whereas in the prime kit it's around 4 nanogram per mil. The reason for this is that the media for the STEM kit is a little different and contains a lower level of serum, and this requires a higher level of growth factor to stimulate angiogenesis. Nevertheless, we are able to measure a pro-angiogenic response over the 4-day assay time course. We also are able to measure inhibition of tube formation, and we can do this in 2 different modes of pharmacologic response using the STEM kit. What I just showed you with growth factor was the development of tubes, and we can look at the inhibition of tube development in neoangiogenic mode. In this mode, growth factor and inhibitor are added at the same point in time 0, and we see in this case with Suramin a concentration-dependent inhibition of tube formation, where 25 and 100 micromolar Suramin completely inhibit VEGF-driven angiogenesis. To look at more complex modeling, we can also look at established mode, where we treat with growth factor for the first 96 to 120 hours of the experiment, allow the networks to establish in what we call established mode, and then add the inhibitor to see if the inhibitor is able to induce regression of these established tubes. Shown on the right is addition of Suramin at 90 hours, where you see a concentration-dependent regression of these VEGF established tubes, where 100 micromolar over the next 3 to 4 days causes a complete regression of these VEGF-driven tubes. We then went on to look at 2 different small molecule inhibitors that are clinically relevant, Sutent and cumbretistatin. Sutent is a small molecule tyrosine kinase inhibitor that targets VEGF receptors as well as PDGFR beta, where cumbretistatin is a microtubule disrupting agent that prevents tubulin polymerization. Again, allowing the networks to develop for the first 90 hours prior to addition of either Sutent or cumbretistatin. When Sutent is added in the presence of fresh VEGF, we see a concentration-dependent regression of these established networks, where 33 and 100 nanomolar Sutent induce regression. The time course is a little bit slower, but by the end of the assay, we see about a 70% reduction using 100 nanomolar Sutent. Further, with cumbretistatin on the right, you see a much more rapid kinetic response in the ability to induce regression, as well as you see at 3.7 nanomolar, it's about an 80% reduction, followed by a little bit of a recovery, but 11 and 33 nanomolar, it is a complete inhibition. Now, I will say we then went back to look at these images, the images at that point, and noticed that certainly at 33 nanomolar, the effect of cumbretistatin seemed to be toxic to the cells, whereas at 3.7 nanomolar, it was not toxic to the ADSCs or ECFCs, as you will see in the next slide. So we can combine the imaging that we have done with the kinetic response to get a very information-rich summary of the experiment. And in looking at side-by-side movies of the Sutent and cumbretistatin response, we can see that while they do both induce regression, this seems to occur via very different mechanisms. Whereas you'll see on the left, Sutent causes a regression of pulling back from the ends of the vascular networks, and you'll see the endothelial cells pulling their ends back toward the middle of the cell, whereas cumbretistatin seems to break the cells apart from the middle, and then you see some recovery by the end of the assay. I'll let this play again so that you can visualize the different mechanisms of Sutent and cumbretistatin. Again, you see Sutent pulling the endothelial cells pulling back from the ends in the presence of Sutent, whereas cumbretistatin, they seem to break apart from the middle. So these are 2 examples of small molecule inhibitors on angiogenesis in our in vitro setting. What happens when we look at large biologics? And here we're looking at anti-VEGF antibody that Lilly has developed against the VEGF receptor, ramucirumab. And what you can see if we look at ramucirumab in neoangiogenic mode, it is able to inhibit the VEGF response in a concentration-dependent manner, where 5 and 10 microgram per mil cause a complete inhibition of developing tubes. Whereas in established mode, when we allow the networks to develop prior to introduction of ramucirumab, we see a concentration-dependent effect, but even as high as 20 microgram per mil, we're only seeing a 30 to 40% reduction of this established vasculature, indicating that there is a portion of the vasculature that is insensitive or resistant to anti-VEGF therapy. While this is nice to see in an in vitro setting, we wanted to confirm this in an in vivo setting, and we did this using a matrigel plug model where we combined these same ADSCs and ECFCs in a matrigel plug and implanted them into mice. We then allowed the vasculature to develop over the next 3 to 6 days, and you see that we do get vascular development, and we are able to quantitate this by staining with CD31. And by day 3, we have vascular development that stabilizes and maintains over the next 3 days. Further, we wanted to test if this vasculature was functional with endogenous the endogenous mouse vascular beds. And we did this by injection of TIR119, which labels blood vessels and is able to investigate blood flow in these in the mice. And you can see at day 4, we start to see TIR119 staining that increases by by day 5 and then again increases further by day 6, indicating that the vasculature that is developing in these mice is able to support blood flow. The arrows on day 6 show that there is some hemorrhaging that is developing. So the vasculature does tend to become leaky, but it is a functional vasculature nonetheless that is able to support blood flow. So what happened when we introduced ramucirumab in this model? Well, if we introduce ramucirumab at the day of implantation and inject the mice with the anti-VEGF receptor antibody, we see almost a complete inhibition of the ability of these cells to develop into vasculature. However, if we allow the networks to develop, over the first 4 days, images shown on the lower 2 panels and quantitation shown on the right 2 bars, we find that the vessels that develop are insensitive to ramucirumab. We see a slight reduction in vasculature similar to our in vitro results, but there's a portion of these vascular beds that are insensitive to ramucirumab treatment. Again, speaking to the ability of these cells to develop resistance to large biologic therapy. So the next question is, can we restore sensitivity of these cells to the large biologic anti-VEGF antibody? Again, on the left, reminding you of the insensitivity that develops, and that using an anti-VEGF antibody, we see a small reduction in the ability of this antibody to regress the established networks. On the right, we're using a combination approach where we treat either with a gamma secretase inhibitor alone, the anti-VEGF antibody alone, or these compounds in combination. The gamma secretase inhibitor that we used was developed by Lilly and has been shown to have an effect on the pericyte endothelial cell interaction with very little effect on actual tube formation. So when we use either of these agents by themselves, the gamma secretase inhibitor or the anti-VEGF antibody, they seem to neutralize the VEGF response when fresh VEGF is added at 96 hours, but they're unable to induce significant tube regression in this model. However, when we add these agents in combination, we see a significant, about 60 to 70% regression of these established networks. Now, did this hold true in the mouse model? So again, we implanted our ADSC-ECFC combination in a matrigel plug in these mice, allowed them to develop for 4 days, and then introduced ramucirumab alone, the GSI alone, or ramucirumab and the GSI in combination. And it was only when these agents were added in combination that we saw a significant, almost complete elimination of this established vasculature, indicating that these cells do develop insensitivity, but this can be restored, presumably by potentially disrupting the pericyte endothelial cell interaction. So here we're studying very complex biology using our in vitro setting that is highly correlative with in vivo pharmacology. And so just to summarize a bit, I mentioned to you both of our kits, the prime kit and the stem kit. They're 2 highly validated co-culture models that are highly correlative with in vivo pharmacology. Again, the prime kit's a combination of fibroblasts with HUVEC, while the stem kit's a combination of ADSCs and ECFCs. These are both human-derived, fully human-derived models, cell-based models that you can choose the best way to analyze your kinetic data. So you take the images over time, and then you can look at max values, you can look at area under the curve, or you can even go back and do single time point analysis, similar, excuse me, similar to what we did with the EC50 generation of the different growth factor responses using the prime kit. Important to note is that no initial time course is required to do this single time point analysis. You can set up the assay, run the full time course, and then choose what time point to do your analysis. Whereas with other endpoint assays, you have to do a time course and then choose a time point there before you actually do your assay. We also can look at complex aspects of pharmacology, whether it be proliferation, migration, differentiation, investigating mechanism of action of different compounds by altering the timing of treatment. And I showed that here using our STEM kit, where we were looking at models of vascular resistance and the ability to use combination therapy to restore sensitivity to large biologic treatment. Further, we offer the Prime Kit and the STEM Kit in multiple different modes, whether that be live cell or cryopreserved with a Prime Kit, cryopreserved only with a STEM Kit, or you can send, if you prefer, we'd be happy to run your compounds here at Esen, set up the kits here at Esen through our Esen Discovery Services program, and generate data and reports of your final experimental results prior to sending them to you. And when we use these in conjunction with our InQSight Zoom, it provides a very information-rich live content approach to studying sophisticated in vitro models, where we can generate mechanistic information, look at paracrine signaling, and generate full temporal resolution of biological events from viable, healthy cells that are unperturbed. I showed you the quantitative plate-based pharmacology that we can generate, as well as the software being very intuitive, simple to use, and the cells, importantly, the cells staying in their native environment in the incubator throughout the duration of the assay. So with that, I'm going to conclude at this point. I thank you for joining us for the webinar. As Jason mentioned, we're now going to open it up to any questions that you may have, and I will answer them the best that I can. And again, please type your comments in the chat box area, and we'll be happy to answer those. Sorry, excuse me. So there's a question here about how the staining is done and if the cells are plated on cover slips. They're actually not plated on cover slips. They're done in the plate where we run the assay in the 96-well format. We fix the cells with paraformaldehyde and permeabilize with Triton X before adding the primary antibody of interest, and then adding the secondary antibody for whatever color we choose. And then in this case, we used a red secondary, placed them in the InQSight Zoom, and then imaged the cells. There's another question about where the cells are derived, how the cells are derived. Our ADSCs are derived from adipose tissue that are separated from during liposuction treatments. It's waste material from the liposuction that is spun down, and the adipose-derived stem cells are generated from areas that are in very close proximity to vascular beds within that tissue. The ECFCs that we use are generated from endothelial or from the umbilical artery cord blood. So the umbilical artery cord blood is isolated, and the cells are grown out, and the isolated colonies that are positive for CD31 are then what develop into the ECFCs. Excuse me. There's another question here about the cost of running assays at your facility, and this depends on the complexity of the study and how many plates we're running. Our single plates, typically the cost of running a single plate at our facility is $5,400 for a single plate. That involves setup of the assay, compound handling, all the dilutions and feedings required. For the STEM kit, it can be 1 or 2 feedings. For the Prime kit, that can be up to 5 feedings. Use of the instrument and imaging, as well as then an extensive data analysis, data report, statistical analysis, and image generation that is sent to you at the end of the assay. Another question is, what is the source of our growth factors? We use basic FGF, FGF2, and the VEGF that we use is VEGF 165. That's the most common VEGF isoform. Is the one that's been studied most extensively, so that's why we've chosen that VEGF isoform. So if there are no more questions at this point, I think we'll conclude. I will say that we will have another webinar next month. I think Jason's going to wrap up here in a second. join us and wrap up in a second, but just to let you know, we will have another webinar next month that will be focused on our migration and invasion studies that we've done at Esen.